Okay, so today I'm going to be talking about antigenic drift and antigenic shift. So antigenic drift is the gradual continuous change of the surface proteins or antigens on a virus, specifically RNA viruses. So these changes happen slowly over time. This is usually what causes new strains of viruses. For my example, I'll just be talking about influenza. So now the way this actually works is that for RNA viruses use RNA-dependent RNA polymerase. And they use that for RNA replication. Now, what RNA polymerase lacks is its ability to proofread. So what this does is it causes more mutations just due to the fact that it lacks the ability to check or detect for mistakes when copying genetic information. So what these mutations do is they alter the antigen shape on the surface of those specific RNA viruses. Why this is so important for the virus and why this is so negative for humans is that these are antigens, right? So what are antigens? Antigens are kind of the binding spots or the binding receptors for antibodies, right? So our body will naturally produce antibodies in response to antigens. If these antigens are constantly changing or have changed over time, these antibodies aren't going to recognize these antigens. Therefore, we're going to lack the immunity for these viruses. Now, antigenic shift is a little bit different, but it's similar in a few ways. Antigenic shift is when the glycoproteins get changed suddenly. So instead of the continuous gradual change over time, antigenic shift is a sudden, more immediate shift in those glycoproteins. This is usually what causes massive outbreaks, especially outbreaks like the swine flu, because we've had little to no time to build up immunity to these viruses because they're so immediate. We end up suffering, and it's really hard to find a vaccine or something that will counteract these antigenic shifts. Now, this isn't a mutation. This is more so genetic reassortment from other influenza viruses coming together. So, for example, swine flu and the Hong Kong virus, those were results of an antigenic shift, because for swine flu specifically, what happened was a pig got infected with a bunch of these different influenza strains, and it recombined into or reassorted into a new virus called the swine flu, which we really had no immunity for at all. I mean, not only are we immune to them due to how sudden they are, but it's because of how different they are, because they're a combination of a bunch of different strains. So they are very different in the strains that we're familiar with. Antigenic shifts are obviously very negative, so are antigenic drifts, but by far, if you had to choose one, shifts would be the worst one. So overall, antigenic drifts are gradual, they are continuous, and they are the change of glycoproteins or surface antigens, usually through mutations. And then antigenic shifts are immediate, sudden changes as a result of genetic reassortment. So